Hi everyone, my name is Deepshikha. I'm a master's student in the Lee Lab, and today I want to talk to you about my project, which is how chromatin is remodeled for DNA damage repair. So all organisms have DNA and are susceptible to DNA damage, like from UV radiation, from the sun, and other cellular damage that can cause cancer and defects in fertility. And so organisms have evolved many different pathways in order to repair this double-stranded break and not lead to toxicity. So how this happens is a little less understood in terms of how the chromatin has to be remodeled in order to allow for the break to be repaired. So here I'm showing you DNA that's wrapped around in gray histone proteins. And this is done to package the DNA into a really small space. And so to allow for the break to be repaired, this chromatin has to be remodeled and pushed away from the break to give more space to DNA repair pathways that can then fix the break, leading to successful gamete formation. And when this goes wrong, it can lead to a lot of fertility defects. And to analyze these defects, we're using our favorite model organism, C. elegans, because it lays 100 embryos like every 4 days. So here we're showing on the y-axis, I'm showing you the percent viable offspring. So out of the embryos laid, how many make it to adulthood? Another way to think about this is percent survival as well. So here in animals without any mutations in wild type, when they're exposed to different levels of the DNA damaging agent, you can see that they're almost always at 90% survival. But these chromatin remodeler mutants, where the chromatin can't be moved away from the double-stranded break, you can see that when there's no DNA damaging agent, they're at still around 90% survival. But as more DNA damaging agent is added, only 70% here are surviving. And here, 30% of the offspring are surviving. And so we want to ask, how does this happen? And so here to do that, we are removing different parts of the protein and asking if there's an effect on their fertility. We're also recreating some residues that we found by sequencing human patients in C. elegans to ask if there's an effect on function of the protein. And with that, I would like to thank my lab for all their support and Kennedy College of Sciences for funding over the summer. And thank you all for listening.